Yeah, thanks for coming to this next edition of the Response Diversity Network Members Seminar Series. So the goal with this series is for us all to kind of get to know each other a little bit better, for people to introduce themselves and their work and find out about what others in the network are doing. And, we kind of shift from different speakers in different time zones. We can host people in kind of any time zone that you're comfortable with. And yeah, the idea is to kind of about once a month have one of these. I think we've got one more speaker after Charlotte, and then we're going to take a little summer break because lots of folks will be on field work and things, and then come back for a few, and then also take a winter break. So maybe 8 months per year, we'll try to aim to have these seminars. And once we finish the recording, I will upload it to YouTube. There's already something like 10, 15 talks up there from the past. Some were in person from a workshop, and some now from this online version of the seminar series. So thank you all for coming live to this edition. And yeah, I'm going to introduce Charlotte. I guess if you could share your slides, Charlotte, that would be great. And then we can do this. So Dr. Charlotte Kunze is a postdoc at the EcoNet lab in iDiv. So Charlotte has just moved quite recently to iDiv. She was in the University of Oldenburg before, doing a PhD with Helmut Hillebrand. And yeah, I actually got to visit Oldenburg once. Charlotte kindly invited me. That was a really nice stay there. We were just trying to work it out. We think we've known each other for about 6 years. So Charlotte was in Trinity College Dublin. She did a collaborative experiment with one of the groups there while I was there as a PhD student, I guess about 6 years ago. And yeah, from the very beginning, I've been remarkably impressed by Charlotte's ability to do what I would call a high throughput experiment, one of these like really, really multifactorial experiments with lots of different things going on, lots of physical labor and work. But she's also gifted at the kind of statistical and conceptual side of things as well, and also has got some cool kind of meta-analysis type papers and things. So yeah, I think I've never actually heard Charlotte speak, so I'm really excited to hear what you have to say about mechanistic insights into the biodiversity stability relationship, Charlotte. So please go ahead whenever you're ready. Thanks.
First of all, thanks, Sam, for the kind introduction. It's a pleasure to be here. Thanks to all of you who zoomed in today. Yeah, as Sam said, I'm going to talk about work that I have been doing mainly during the last 4 years. So doing my PhD, basically. Right. As we all become increasingly aware, human activities are profoundly affecting the Earth biosphere in unprecedented ways. This includes changes to the biotic environment, such as invasive species, but also to the abiotic environment, such as floods, hurricanes, or heat waves. Such alterations to the biotic or abiotic environment that affect the structure and dynamics of ecosystems are termed disturbances. Such disturbances can be briefly categorized into 3 main types. That is, fluctuations, here displayed as the disturbance magnitude over time, so gradually, stochastically, or continuously varying environments, such as temperature variations or changes in water levels. Pulse disturbances that are short-term extreme events, such as floods, hurricanes, or heat waves, so an isolated event in time. And press disturbances that are a continuous lasting stress, such as an invasive species or climate change-induced changes. In nature, however, disturbances are often way more complex and differ in magnitude, duration, frequency, and their spatial and temporal extent. As you can see here, And some of the examples by Ian Donoghue et al. From 2016. So here, for instance, the global sea surface temperature anomaly over time. So given that disturbances are often way more complex than the simple categorization might suggest, the question then arises, how can ecosystems maintain their stability? When I talk about stability, I'm referring to the concept of ecological stability. That is a multidimensional concept that tries to capture the system's dynamics and responses to disturbances. And it's called multidimensional because it comprises multiple dimensions. So to describe a system's ability to recover and resist change, we can use different stability metrics. So here, following a disturbance, we'll see likely a deviation of our system state, and we can measure the ability to withstand this disturbance using resistance, A, the ability to recover from the disturbance, that is B here, and the amount of variation along this recovery trend, that is called temporal variability. Finally, we can then measure the amount of final deviation from control conditions, here called recovery D. All of these stability metrics are measured relative to an undisturbed control to get an idea how much variation is really because of the disturbance and not because of seasonal or naturally occurring variation. All of these stability metrics can then be summarized into one single integrative metric, and that's the OEV index, or Overall Ecological Vulnerability Index. It is based on the area under the curve of the relative response of our system to an undisturbed control, and the interpretation is quite easy. So if I have a large OEV index, it means I have large instability. We can further distinguish between positive and negative deviations from control condition, which gives us an idea if our system is negatively or positively affected from our disturbance. We can further distinguish between compositional stability and functional stability. Compositional stability here describes the stability of species composition, whereas functional stability describes the stability of functional properties of our community, meaning productivity, biomass, or abundance. In fluctuating environments, compositional stability and functional stability are then negatively correlated, or tend to be negatively correlated, because a change in species composition is essential to maintain functional stability in the face of disturbance. This is also why biodiversity is then positively linked to functional stability, as biodiversity in our community promotes functional stability by allowing for higher compositional turnover. What remains unclear so far, however, is how individual species influence community stability. I will aim to answer that question using 3 different parts, and in the first part, I'm going to talk about a framework on how to partition species contributions to stability. In the second part, I'm going to talk about the role of response diversity for community stability. And in the third part, I'm going to introduce the net effect of biodiversity on stability. Right. So in a framework on how to quantify species contributions to stability, we can use that species-specific responses differ. Species can change in biomass or abundance following a disturbance, as displayed here, the increase in magnia abundance, Daphnia magna abundance following disturbance, but they can also change relative to the rest of the community. So in relative terms, in proportion. This leads us then to a 2-dimensional framework based on species change in biomass and species change in proportion. So based on species change in biomass, we can then estimate their absolute contribution to stability here on the y-axis, whereas based on species change in proportion, we can estimate their relative contribution to stability. In a community, we will then find species which suffer because of the disturbance here in red, so they decrease in biomass and in proportion and are contributing negatively to stability. We'll find species which suffer, but less so than others, meaning that they are decreasing in biomass, but still increasing in proportion. We'll find species which benefit and increase in biomass and in proportions, so they contribute positively. And we will find, or maybe not find, species which benefit but less so than others. And as you may can imagine, that's a very rare case. So species can contribute to stability in two different ways. That is by being tolerant to the disturbance and increasing in biomass and in proportion, or by being less sensitive than other species and contributing positively in relative terms. To test our framework, we did some Lotka-Volterra competition model simulations and introduced different sensitivity scenarios and disturbance types. So here you can see some exemplary model runs where the different facets indicate the different disturbance types. And we distinguished between a press disturbance, that is a continuous biomass reduction, a post disturbance, that was a sudden 50% biomass removal, and a combination of those two. We further distinguished between different sensitivity scenarios, where in the first raw, all species were equally sensitive and got reduced following the disturbance, whereas for the second and third raw, where there's always one different species, we can see that one species in the second raw is increasing following the disturbance, here in blue, while in the third raw there's one more sensitive species which gets reduced in biomass and is displayed here in red. So if we then apply our framework and look at species' absolute contribution and their relative contribution, here for the case where all species are equally sensitive, what we can see very nicely is that our framework picks up that all species are reduced in biomass, here the red dots. However, proportional change is not present. If you then look at the two cases where there's one more resistant species and one more sensitive species, we can see that really here in blue, the one more resistant species is displayed in panel 1, whereas the one more sensitive species is displayed in panel 3. So we can really see that our framework seems to be able to identify the stabilizing and destabilizing species here, and that species benefit and suffer according to our sensitivity scenarios. As a second step, we wanted to test this framework using some empirical data. And here we used a published multi-site mesocosm experiment that was conducted in 5 lakes and 2 seasons all over Sweden. Here on the right side, you can see an exemplary setup, basically. We looked at zooplankton biomass over time and looked at different treatments, which consisted of an undisturbed control, a press disturbance, so a light reduction over time, a pulse disturbance, which was a short-term fish addition of 7 days, where an unselective grazer got added to the system, and a combination of the pulse and press disturbance. So we had, again, the fish addition with the light reduction. When we then applied our framework, and I removed the points here for better visibility, we can have a look on the absolute contribution to stability on the y-axis again, and on our relative contribution to stability on the x-axis. The different facets indicate the different disturbance types. The different legs are indicated here as different draws. So here's what we find, and the different zooplankton taxa are indicated by different colors. What I want you to take away from this very messy graph is that we find similar patterns. So again, we see that there are species that benefit following the disturbance, and again, species that suffer because of the disturbance. But also we see a high context dependency. So just by looking at the color distribution, we can say that very rarely one species contributes in the same way, in the same lake, and in the same disturbance types. If we zoom in to one lake in particular, Lake Fjärsfjön, here again on our y-axis, the absolute contribution to stability, on the x-axis, or relative contribution to stability, where the different facets indicate the different disturbance types again. what we can see if we zoom in a little bit more and look at one species in particular, the large dominant zooplankton species Thermocyclops, is that there are very clear differences among disturbance types. So for the press disturbance, Thermocyclops is contributing positively and is increasing in biomass and in proportion. Whereas for disturbances that involve a pulse disturbance, we see that Thermocyclops here and here seem to be contributing only in relative terms. If we then look at species dominance, which is indicated here by point size, we can see that rare species, which are less dominant, seem to be increasing following the disturbance, whereas dominant species, here the large point sizes, seem to be decreasing following disturbance. So we do see some compensatory dynamics. So to summarize this first part, we can say that the identity of the destabilizing species here differs among seasons, lakes, and disturbance types, and that there are compensatory dynamics happening, meaning that even species that are presently rare can play a key role in the future. Also, we find that species contributions to stability are highly diverse and context dependent. Right. So the question then arises, can we use this diversity in species responses to predict community stability? The phrase response diversity was established by Elmquist in 2003 and briefly describes the variability in species responses to environmental change. It got turned into two consecutive response diversity metrics, dissimilarity, so the spread of species responses, and divergence, which describes the direction of species responses, in 2003. On the right side, you can see two examples where in the first row, there's the example of low response diversity, and here species responses are narrowly spread along an environmental gradient. So species respond quite similar to the same environment. Whereas for the high response diversity case, species responses are far spread along an environmental gradient and also differ in direction for a given environment. This response diversity, however, was mainly conceptualized in fluctuating environments and using species responses in isolation. However, as we just heard in the beginning, there are different disturbances, not just fluctuations, but also pulse and press perturbations. So in the next part, I'm going to talk about how we can conceptualize response diversity in a pulse disturbance context. So here we can see species fundamental responses as their intrinsic growth rates in isolation over an environmental gradient. Here are displayed 4 exemplary species, which differ in their response to the environmental gradient. In a pulse disturbance context, we would then expect that species responses remain somewhat constant or reflect their response for the given environment, and which then increase or decrease following the disturbance. So, for instance, here, the species displayed in green is experiencing an increase in its intrinsic growth rate following the disturbance, which, however, then gets reduced and is returning to the pre-disturbance conditions. So we can then actually calculate an intrinsic growth rate effect on those species. Using this intrinsic growth rate effect, we can calculate the different response diversity metrics. That is response dissimilarity, response divergence, and additionally, we can look at the mean response here. So what we see if we look at the spread of species responses, so how dissimilar species responses are, we see that they're somewhat dissimilar. So we would have a positive or slightly enhanced dissimilarity. Also, species responses differ in their direction. So we would expect a high divergence and a positive mean response. However, very often in empirical ecology, we don't have fundamental responses. So in the next step, we looked at species realized responses. So species net growth rates in the community here in a disturbed community relative to an undisturbed control community. And if you then look at species-specific responses in the community relative to an undisturbed control, we can actually calculate their absolute contribution to stability and use this as a measure for realized species responses. So here in the bottom row, you can then see the different species responses to a pulse disturbance, which vary then if we look at the response diversity metrics again in dissimilarity. However, the direction is quite similar. So we would expect a lower divergence and a negative mean response. As a second step, we tested this conceptually and used a Lotka-Volterra species competition model, this time with temperature-dependent birth and death rates. Here you can see an exemplary model run on the right side, where species responses differed to our pulse disturbance. As a pulse disturbance, we introduced a temperature drop and also looked at different interaction strength. As a second step, we then looked at species fundamental responses in isolation, as well as their realized responses in the community. We'll start here with the fundamental responses, so species responses in isolation. And here you can see community stability on the y-axis as a function of the mean fundamental response. The 0, 0 line indicates here that our community is stable, and the different rows, so the upper panel shows there is no interaction between species, and the lower row that there are strong interactions between species. So what we can see if we look at the first graph is that there's a monotonic relationship between community stability and the mean response. Whereas if we increase species interactions, we see a shift towards a more positive mean response for a community to be stable or remain stable. If we look at the fundamental response dissimilarity, then we see that for a community to be stable, there needs to be high response dissimilarity. Whereas if we have strong species interactions, we see a shift towards intermediate response dissimilarity, meaning that response dissimilarity becomes less important for communities with interactions. And if we look at divergence, so at the direction of species responses, we can see very similar patterns. So again, if there is a community without interactions prevailing, we need high diversity in species responses in their direction for a community to be stable, whereas for communities with strong interactions, this clear relationship vanishes. As a next step, we can then look at species realized responses, so their responses in community. Again, on the y-axis, we have our community stability as a function, this time as the mean realized response. And for communities without interactions, we see the same monotonic relationship between community stability and the mean response. Whereas if we increase interaction strength, we see again the shift towards a more positive mean to maintain stability. However, also a lot more heterogeneity. If we then look at response dissimilarity, we see that if there's no species interactions, stability again requires high response dissimilarity. And if we increase interaction strength, a shift towards lower response dissimilarity to maintain stability. Lastly, we can look at the realized response divergence. And while the community without interaction shows the same patterns as before, if we increase interaction strength here, we can see that the relationship between realized response divergence and community stability completely vanishes, basically. As a next step, we wanted to test this again using some empirical data, this time from a meta-analysis on pulse disturbances. And we used here 98 studies from different systems. Again, we looked at community stability that was measured as the OEV index and looked at species realized responses as we did not have fundamental species responses for our species in the meta-analysis. So here's what we find. On the y-axis, we have our community stability, and the x-axis, again, our mean realized response. And we see actually the same monotonic relationship between community stability and the mean response as we saw in our model simulations. For response dissimilarity and response divergence, however, we see that response diversity here seems not to promote community stability. So there's no clear relationship. This tells us then that in natural communities with unknown interaction strength, community stability does not emerge from response diversity. So contrastingly to fluctuating environments, pulse disturbances very often depend on uniform resistance across species instead of a large variety of species responses to maintain stability. So to summarize this, we can say that response diversity is less effective in a pulse disturbance context than it is for fluctuating environments, and that stability depends on uniform resistance across species. So there's always one good response for species to maintain their productivity or performance in the community following disturbance. Also, we saw that the importance of response diversity for community stability depends on the interaction strength. And that's with more interaction strength, those stronger species interactions, there is a more positive mean response or a lower response diversity required to buffer disturbance effects. Which then suggests that not only the structure of species responses, but also the nature of disturbances critically shape ecological stability. So now that we talked about how species responses are influencing community stability, we can next ask, what effect does biodiversity has here, or how much stability emerges as an effect of biodiversity? The related question, how biodiversity affects ecosystem functioning, thereby has benefited tremendously from calculating an expected biomass yield based on species-specific responses in monoculture or species-specific biomass yields in monoculture, and comparing this predicted biomass yield with what was actually observed in the mixture. So by taking the difference between what is observed and what is predicted based on monoculture, Hector and Leroux established this net biodiversity effect on ecosystem functioning. To transfer this to stability, we can again look at species-specific responses in monoculture over time. And by looking at species responses in the disturbed environment, we can calculate what we would expect how our mixture would perform in a disturbed environment by taking the difference to the control and calculating the area under the curve. We can then calculate actually an expected stability based on species performance in monoculture. We can then compare our expected stability with what we actually observe in the mixture in our disturbed treatments. Here, the red dotted line with the D, and calculate the stability of this disturbed mixture. And by comparing them or observed and expected stabilities, we can come up with this net biodiversity effect on stability, which in this case is negative, as the observed stability is lower than we would have expected from monoculture. Of course, the net biodiversity effect on stability can also be positive, and that's the case if our mixture performs better in the disturbed treatment than we would have expected. So here we see actually greater stability than we would have expected based on monoculture, means that biodiversity has a positive effect on stability here. To test this framework, we conducted a phytoplankton experiment, a microcosm experiment using 5 different phytoplankton species that were isolated from the North Sea and that are abbreviated in the following based on their first letter. We combined those species in different species richness combinations. So we had monocultures of each species, 2 species in combinations, 4 species, and 5 species in combinations, and then exposed all of these species communities to different temperature treatments that comprised a constant control, a diurnal temperature fluctuation, a temperature increase, and a combination of temperature increase and fluctuation. We chose all of those temperature treatments to have a somewhat negative effect on phytoplankton communities on average, either because of the temperatures that are reached or because of the fluctuation frequency. Here's what we find. So here on the y-axis, we have our net biodiversity effect on stability, and on the x-axis, we have our species richness, where the different treatments are indicated by different shapes and colors. First of all, what we see is that the observed stability differed from expected stability, that at least overall, we see a more or less positive net biodiversity effect on stability. However, this net biodiversity effect on stability decreased non-linearly with increasing species richness. And there are very clear differences between the disturbance regimes. So for instance, if we look at the temperature increase, here the blue triangles, what we can see is that for 4 species in combinations, the NBS even turned negative. And that might be likely because of the temperature increase treatment, as it is known to enhance competition between phytoplankton species. If we zoom in a little bit more into the 2 and 4 species communities here, what we can see is that there's actually a lot of heterogeneity in the NBS among richness levels, which suggests that there are more diverse community responses under different disturbance regimes, but also that rather than having a uniform response across the entire community, there is an idiosyncratic compositional effect on the net biodiversity effect on stability. So in summary, we can say that stability emerges as a community property that is influenced not only by species richness, but also by species identity and their associated traits, and that biodiversity can have both a stabilizing and destabilizing effect on stability, depending on the species in combination. Coming back to our initial question, how do single species influence community stability? In the first part, I introduced a framework on how we can partition species contribution to stability and showed that species contributions are actually very context dependent. But also we can see that there are compensatory dynamics happening because of different disturbances, which also influence how our community might be stable or if our community is stable following disturbance. In the second part, I then looked at response diversity as a determinant of community stability, and I found that the importance of response diversity depends on the disturbance type and interaction strength. And in the third part, we looked at the net biodiversity effect on stability and found that stability differs among disturbance regimes and with community composition, so how well species traits match the actual environment. This suggests that for maintaining ecosystem functioning under environmental change, it is essential to maintain high levels of biodiversity, and that the loss of species may have unforeseen and severe consequences for ecosystems under climate change. With that, I want to thank you all for your attention, my lab and my co-authors for their support. Thank you.
Fantastic, Charlotte. Thank you so much for that. That was great. I guess if you want to stop sharing your slides, and then I can maybe see if people have questions. You can, as I said, you feel free to write in the chat if you don't want to kind of be recorded on Zoom and put on YouTube later. Or you can raise your hands and ask a question. I think Jeff has one. Please go ahead.
Hey, thanks. You hear me okay?
Yeah.
Yeah, thanks, Charlotte. It's nice to hear you present your work. I've been following what you've been doing recently, actually. I had maybe a super basic question about the philosophy behind the start of your talk, where you partition species contribution to stability with this change in biomass versus change in proportion. And that, I mean, it seems intuitive as a description of species responses, but I didn't really understand why you say it's a contribution. Like, I didn't really get the underlying argument by which, yeah, the classification into other species contributes that versus another contributes something else. Like what are they contributing to? What's the sum total where you see the partitioning pop up?
Thanks for your question. Very interesting. So I guess it depends on your definition of stability. For a community to be stable, we can distinguish how much species biomass then contributes just in absolute terms and how much it contributes in relative terms. So in comparison to the rest of the community. So if we assume that we have a what is it called? A biomass reducing disturbance. So a very typical disturbance. For our community to be stable, I need species to contribute biomass and to balance out the negative responses or the reduction in biomass of other species. That's the argument behind that. So that if we have a stable community biomass to reach that again, following disturbance, species need to contribute biomass or just be less reduced in biomass compared to other species.
But then why not just look at the relative biomass change?
Because very often, if you only look at the relative biomass change, species might be contributing positively, but in absolute terms, it's not enough. So only because my species is being less affected by the other species or is increasing in relative terms, the amount of biomass that is lost by other species does not balance out with what is gained. So compensatory dynamics are not always perfect. This is why we need this absolute access.
Yeah, I understand, but the absolute axis would be just the total biomass change. You don't need to repartition it by species, because then it's redundant, no?
I wouldn't say it's redundant. I think it's necessary that we see how much each species contributes in absolute terms based on how much one species change, and then also relative to the rest of the community, so proportionally. And in the second part, we actually looked at how this absolute contribution to stability then aligns with community stability. But yeah.
Okay. Maybe I'll think about it a bit more. Thanks.
Thanks for your question.
Oh yeah, Francesco's room has a chat, a question.
Thanks very much, Charlie. It's from me. Hi.
Hey, Owen.
In the last third of the talk, you introduced this what was it? Net biodiversity effect on stability, NBES, and that's calculated as the Well, let's just go. And it kind of partitions out what one would expect if one just took the monoculture information and added it up, and looked at the stability difference of the sum of the monocultures from the stability of what was actually observed when the species were in a community together. for the expected mixture versus the observed mixture. And you kind of drew this analogy with the Roe and Hector partition, where they partitioned the total biodiversity effect into what they called the selection partition, the selection effect, and the complementarity effect. So they were proposing, or they created this partition that was designed to partition between two different processes or mechanisms of biodiversity influencing function. One mechanism being selection, it's covariance between traits and abundance effectively, and the other being complementarity, niche complementarity, for example. Now, in your, if I call it a partition, I'm interested in how, and if you see that as partitioning between any of the, or which of the processes or mechanisms that can lead biodiversity to stabilize communities and ecosystems. What's that mapping, if any, between your partitions and the processes, the stabilizing processes that have been proposed, for example?
Right, very good question. So this net biodiversity effect on stability, we developed this in a relative context, I want to call it. So it's always relative to an undisturbed control. So we can't really distinguish between complementarity and selection effect because those are in an absolute area. Whereas if we look at the relative, or this is a relative ratio basically in our calculation. What we could do is, of course, look at how species contribute to stability and then merge this with the net biodiversity effect on stability. But we are not able to really distinguish those two different effects that you were just mentioning, yeah.
Well, I mean, when I think about the processes that could lead biodiversity to stabilize its processes, I think about things such as asynchrony in population dynamics. And I think, yeah, and so I wonder about how asynchrony could go into one or both of the components, kind of like the net and the gross here.
Right. We haven't looked at asynchrony yet. What we did, however, is that we looked how species traits influence the net biodiversity effect on stability. So we kind of did the other approach where we looked at if species are there and if the traits are there, is the NBS more likely positive or negative? Because it really depends on the disturbance regime you're looking at, right? But yeah, asynchrony as such, we haven't measured yet. Yeah.
Thank you.
Great. There's a question in the chat that I'm going to read to you from Shang Yang, who's at iDiv, I believe. He says, I wonder what thanks for an interesting and nice talk. I wonder what's the net biodiversity effect on the different individual stability components in your experiment. So he says, you found no directional effect on the overall stability, but what about the individual stability components? So I assume resistance, resilience, and so on. Like many studies found positive effects on temporal variability at the community level, for example.
Yeah, that's a very interesting question. So when we looked at the net biodiversity effect on stability for resistance and resilience, we saw that more diverse communities, so communities with higher species richness, were less resistant than communities with lower species richness. So we actually saw that they differ, whereas for temporal stability, we saw the reverse trend. So it really seems to depend how many species are in a community, what kind of stabilizing mechanisms take place. So if it's resistance or resilience. Yeah, very good question.
Great. Any other questions for Charlotte? You can also write in the chat if you want. I think I have something written down. What was it? Oh yeah, I was wondering about the so when we're talking about the temperature effects on net the net biodiversity effect on stability. I was wondering about dominance. Is dominance already kind of accounted for because of the way that you're thinking about relative changes, or could, for example, one species that prefers certain temperatures really drive that whole net effect still? Sorry if I've misunderstood something there.
I know that's a good question. I mean, we haven't looked at dominance in particular. We looked at how species thermal preferences drive the
Right.
influence on the grand mean. But of course, if there's one species which is highly dominant or highly competitive, it might actually drive this. Yeah.
Okay, great. Thanks. Last chance for questions from anyone in the Zoom. If not, then let's thank Charlotte again for what was a really interesting talk. Thank you very much, Charlotte.